every hand in the room is going to
Yeah,
go
I
up,
agree.
right? It matters a lot. Um, and so then what is cancer? It used to be one thing when we didn't know much about it,
Mm.
but it turns out it's lots of things. And then the world has changed in terms of how we understand it. It used to be that the sort of way we would think about cancer was, where did it arise? Is it a breast cancer? Is it a colon cancer? Is it a brain cancer? And interestingly, that kind of morphologic, visual body part description remains the standard. When you ask people what kind of cancer is, that's usually the answer that they're going to give you. But the ability to interrogate the genes of human beings and their tumors has begun to change the game and provide a completely different level of insight into what cancer actually is. Because
Hmm.
there are certain commonalities in how cancers come about that might make a cancer of one particular origin more like a cancer of a different type based upon its genes than those that actually look the same under the microscope and might need to be treated differently. So we're beginning to start seeing cancers in a different way being driven by alterations in their genes. But then that's raised yet another complexity, which is it isn't just a deterministic sense of something going wrong with our genes. You can't disentangle our genes from our
Yeah,
environments, and there's
yeah.
an interplay with the environment. So it's turning out that cancer is a very complex set of diseases that has at least one commonality. And that commonality is the fact that the cells in the body are supposed to listen to the central command. And in cancer, they have stopped listening to the central command and have gone rogue. That's the commonality. The reasons that they do it differ sometimes within an individual. So
Mm.
that creates a more complex landscape in terms of thinking about how do you diagnose this and how do you treat it. However, the amount of knowledge has moved so quickly, in no small part because of technological advances, that need to be treated differently. So we're beginning to start seeing cancers in a different way being driven by alterations in their genes. But then that's raised yet another complexity, which is it isn't just a deterministic sense of something going wrong with our genes. You can't disentangle our genes from our
Yeah,
environments, and there's
yeah.
an interplay with the environment. So it's turning out that cancer is a very complex set of diseases that has at least one commonality. And that commonality is the fact that the cells in the body are supposed to listen to the central command. And in cancer, they have stopped listening to the central command and have gone rogue. That's the commonality. The reasons that they do it differ sometimes within an individual. So
Mm.
that creates a more complex landscape in terms of thinking about how do you diagnose this and how do you treat it. However, the amount of knowledge has moved so quickly, in no small part because of technological advances, that there's a much deeper understanding. And there are cancers that arise in certain ways and cancers that arise in others. Some of the simplest ones are, for example, and I don't mean to say that they're simple,
Sure.
but for example, virally developed cancers like human papillomavirus or cancers that arise from hepatitis. We understand those. Certain cancers we know come from germline, or meaning inherited defects like breast cancer and ovarian cancer that come from BRCA lesions, or Lynch syndrome with mutations that happen in colon cancer. But most cancers seem to arise out of the blue, and we've got a much deeper understanding of the genetic basis of those, and increasingly how the environments might interact
Mm.
with the genetics to cause those cancers.
I can ask you what might be a really naive question. There's going to be a few of these. Why are there some cancers that are incredibly aggressive and some that are not? And
Right.
there are malignant tumors, benign tumors. There are um,
Yeah.
certain cancers with high mortality rates, one with very low mortality rates. Is that a really stupid question? But
It's not a stupid question, and it's one that still perplexes
mm,
and challenges many of us in the field. But let's try to break it down. So, why are some cancers so much more aggressive than other cancers? Well, one reason is that cancers that are harder to detect earlier are more difficult to treat.
Hmm.
So if you look at, for example, glioblastoma, the most common brain cancer, or pancreatic cancer, you really can't detect those things earlier. So the mortality rates are very poor.
Because
And
they've progressed so much more
by the
aggressively.
time
Right.
that you find them,
Got
which
you.
really leads into a discussion about early detection,
Sure.
right?
Yeah.
But if you look at certain cancers where we screen, for example, things like colonoscopies and things like that,
Prostate.
right? Prostate, uh, people in general do better, but that's not the whole story. There's intrinsic biology. So what we discovered is uniformly associated with poor outcome in patients because the biology is so different in those cancers. So there are both, I would say, external reasons in terms of when we catch those cancers, and there are internal reasons in terms of the biology itself that makes them more aggressive.
Let's talk about that because, I was a little familiar with your work. Obviously, we've worked together for a couple of years now, but this concept that, there's more at play with your DNA than we believe when it comes to the sort of growth and aggressiveness of the cancer itself, and ecDNA, I believe I'm saying that
You're
correctly.
saying it right.
I was kind of fascinated about the idea that not only does that sort of add fuel to the fire, but it kind of tears down the smoke alarm as well. Can you maybe unpack that and help the audience understand what
I
that means?
can. What you're referring to, this ecDNA, is called extrachromosomal DNA. And let me give a backstory to what it means, how we found it, and what we've learned about it. So the backstory is, when we think about genes, we think about chromosomes. I mean, it's imprinted on our heads, right? You look at a box of 23andMe,
Yeah.
we're always thinking chromosomes,
Yeah.
right? That's how genes are inherited. That's how genetic information goes from parents to their children, or when a cell divides, it goes on chromosomes. And so that's really the basis of pretty much everything we know about human genetics. So anything that is not within that line of thinking is kind of banished.
Yeah.
We wouldn't even think about it. And what we discovered in about close to 20% of all people with cancer, and probably more in those in which it's later stage and metastatic, is that the cancer-causing genes have jumped off chromosomes onto circular pieces of DNA. So why does that matter? It's because they've untethered themselves from the normal constraints of how genetic information is passed. So let me give an example about why it actually matters, and then maybe why we missed it until now. And interestingly, it had been seen but not understood. So let's go back and say, what does this actually mean? So let's just say you had 3 copies of a gene on chromosomes. When the cell was getting ready to divide, normally what happens is you duplicate it. So you got 6, and each daughter cell would get 3. But these circular pieces of DNA don't have the same machinery that chromosomes have. So those 3 become 6, but instead of going 3 and 3, it could be 6 and 0, or 5 and 1, or 4 and 2, or 3 and 3. So think about this on one thing. You're supposed to get 2 copies, but now a cell got 4, right? And now if that's advantageous, that 4 is going to become 8.
Sure.
And now you keep getting the spread. So 2 things are happening at the same time. You're making extra copies of a gene that's promoting cancer, and you're creating variation, which Darwin taught us is the fuel for evolution. You have a landscape to select. The likelihood that a cell that is more fit is sitting right there. So it's almost as if we can learn something from nature, because in fact, that's exactly what bacteria do. Bacteria have circular genomes, and they have plasmids. And daughter cells, each daughter is a unique organism, and daughter cells don't have to have the same DNA as mother cells. And consequently, the species evolves very quickly. That's why we struggle with bacterial antibiotic resistance. But this would have been unthinkable in human cancer. Wait, they're supposed to have chromosomes. It is as if cancer pulled a trick. It hijacked an ancient system to allow rapid genome change. And that's why cancers become resistant to our treatments so quickly.
So is it effectively, as the cells divide, they're just rolling the dice?
Exactly. Every cell division is a dice roll.
皆さんこんにちは，卡林基です。大家好，我是来自日本的卡林基老师。你看看今天的打扮这么的轻松。